So welcome to Ag Canada Land. So my name is Charles Geddes, and I am the weed scientist here working with Ag Canada based out of Lethbridge. And so today we're going to be talking about Kochia for about the next half an hour or so. So just a little bit of a background, I guess. First I'm going to start off, go through a little bit of history on Kochia. Obviously, the reason why we're talking about it is because it's a huge problem in this area, and we're having a lot of difficulties with herbicide resistance. Okay, so I'm going to talk a little bit about herbicide resistance. Then we're going to talk about the general biology of the weed, and from there we'll work our way into a series of experiments. So currently, the reason why I'm talking about Kochia is I'm leading a prairie-wide project through the Integrated Crop and Agronomy Cluster. And this project was funded by Western Grains Research Foundation, as well as Manitoba Pulse and Soybean Growers and Manitoba Wheat and Barley Growers Association, in addition to funds that were matched through Ag Canada. So the project is focused solely on figuring out how to manage herbicide-resistant kochia, with a heavy focus on glyphosate-resistant kochia. So, does anyone know what groups do we have herbicide resistance to in kochia? Does anyone know? 2, 4, and 9. Okay, so that's exactly it. So back there in the States, actually, they also have four-way resistance. So in the States, the first type of resistance that was found in kochia was back in I think it was 1978, where they found resistance to atrazine, which is a group 5 herbicide. And they found that population was cross-resistant to a range of the group 5 herbicides. And then only after that, they actually found ALS inhibitor or acetylactate synthase inhibitor resistance, which is group 2. So that was originally found in Kansas and Iowa sort of area in the United States. So that would have been back in about 1987 in the States. And then 1 year later, we actually found that in multiple provinces here in Western Canada. So we've had ALS inhibitor resistance in Western Canada since 1989. And so that was the first type of resistance that we've found here in Western Canada. And from there, ALS inhibitor resistance spread very quickly. So over the course of about 2 decades, ALS inhibitor resistance in kochia spread to the point that now we consider every population in Western Canada resistant to group 2 herbicides. So it's to the point where we do a large amount of screening for herbicide resistance, and it's very difficult for us to even find a susceptible check population for our herbicide resistance screening. So that's how widespread group 2 resistance is in kochia. So from there, does anyone know when they found glyphosate resistance in kochia in the States? It was in 2007 in the States. They found glyphosate resistance in Kansas. And then in Western Canada here, we found glyphosate resistance in 2011. So there's kind of this recurring pattern. We usually find this type of resistance in the States, and then we'll see it up here in Western Canada as well. The question is, are we selecting for it here, or is it actually transferring across the border? And we're not really sure, but I do think that a large portion of the resistance that we're seeing is actually being selected for in our cropping systems and not actually transferring up. So we've had glyphosate resistance in kochia since 2011, and the first populations were actually found in chemical fallow fields that were just south of Lethbridge here in Warner County. And from there, in 2012, they did a survey of Alberta to see where this type of resistance occurred. And that survey showed that about 5% of the populations, they collected about 300 populations across Alberta, and about 5% of the populations at that time in 2012 were resistant to glyphosate. And then they also did another survey in 2013 of Saskatchewan and Manitoba, which also in those provinces showed that about 5% of the populations were resistant to glyphosate at that time. The the real question was, first we found resistance out here in Alberta, but we know that kochia is a tumbleweed, right? And are those populations, are we transferring resistance to the other provinces through the tumbleweed mechanism, right? So are these plants blowing and tumbling westward, and that's how we're seeing the resistance move westward? But that we really don't think that that's actually the case. And the main thing was because the populations that were found in Manitoba were actually found in different types of cropping systems. They were mainly found in glyphosate-resistant soybean and corn, whereas out here they started in chem fallow, and now they've expanded into a wide range of crops, most of the crops that we grow. So that's kind of a background on most of the herbicide resistance that we have in kochia. The new type of resistance that we're finding is resistance to group 4 or the synthetic auxins. And I'm really interested in this type of resistance because group 4 resistance will have quite a bit of implications for many of our cropping systems. A lot of the really efficacious herbicides that we rely on in our small grain cereals are group 4 herbicides. So a lot of those group 4 chemistries historically have been very effective at managing kochia, but it will mean drastic changes if we do see widespread resistance to the group 4 herbicides in Alberta. So the final thing that I want to say about the distribution of herbicide resistance, the survey that was done in 2012 was just repeated 5 years later in 2017. And again, this survey was led by Hugh Becky out of Ag Canada in Saskatoon. And they collected over 300 populations from southern Alberta. And does anyone have a guess on the level of resistance? So we were at 5%, say 5 years ago. Does anyone know what we're at now? 3%? I wish. 93. So we're at 50% now. So over the course of 5 years in Alberta, we saw glyphosate resistance increase from 5% of the populations to the point that now half of the populations out there are resistant to glyphosate. So that is a very quick spread of resistance. And our goal is to try and slow this spread, but also try and figure out how to manage these herbicide-resistant biotypes when they are out in your field. So in addition to glyphosate in that survey, they also found dicamba resistance in 18% of the populations in Alberta now. So that was not found in any previous surveys. And 10% of those populations were resistant to the 3 herbicide modes of action. So the group 2s, the group 4 being dicamba, and glyphosate, which is a group 9. So 10% of the populations in Alberta are triple resistant. Some of the work that we're looking at right now, we're wanting to see if these group 4 resistant biotypes are cross-resistant, meaning resistant to multiple active ingredients within the group 4 mode of action. Because we do use dicamba in our cropping systems, and especially say with Extend soybean moving into Western Canada, there will be some fairly large implications with resistance to dicamba and glyphosate. But we actually rely on another active ingredient, fluroxapyr, a lot more, especially in the small grain cereals like barley and spring wheat. So what we're trying to do is characterize resistance to both dicamba and fluroxapyr to see if these populations are resistant to multiple active ingredients within that mode of action or not, because fluroxapyr will have greater implications here in Alberta for sure. And some recent, I guess, results that we've had in those screening efforts, we have found there is fluroxapyr resistance in Alberta and Saskatchewan. We don't know the extent as of yet, but we are working on that. The way that we commonly try and characterize or identify whether a plant is resistant or not is what's called a dose response experiment. So we have these multiple populations, and we subject the populations to a range of doses of the herbicide in the greenhouse. And we compare the response of those populations relative to multiple susceptible controls as well as resistant controls. So we can tell, based on the above ground biomass that these plants produce at the different rates of the herbicide, whether the plants are resistant or not. So to do that, we pull out a value called the GR50, or the growth rate 50 value, which is the dose of that herbicide that's required to reduce biomass of that population by 50%. And for what we saw for fluroxypyr in our most recent testing was that the GR50 value ranged from, it would have been 2 grams of active ingredient per hectare, which would be our most susceptible control. The susceptibles range from about 2 to 40 grams, but our most resistant kochia population was actually about 950 grams. So it's way more resistant, okay? So our goal is to characterize those 300 populations in Alberta to see the extent of fluroxypyr resistance as well. Eating our plots. Okay, so that's kind of a history of herbicide resistance in kochia. Moving on to some of the biology. So why do we see resistance showing up in kochia so rapidly, right? So kochia actually has many of the biological characteristics that we consider in a weed to be highly selective for resistance. So it's, first of all, the populations are very genetically diverse, or they're very diverse. So some recent research has shown that populations, say in a field like this, so this 28 acres that's assigned to our research program, we have multiple biotypes out here. You can see them by just looking at them. The genetic diversity within this field is as diverse as among this field and, say, a field in Manitoba. So it's very diverse. All of the populations are very diverse. In addition to that, it's very drought and saline tolerant, which is why it tends to grow so well in this area. And we also, it tends to thrive in those areas where our crop really does not, right? So you often see kochia showing up in those low-lying kind of saline areas, which makes it very easy for that plant to outcompete everything else in that area, right? So kochia, it's what we would call, it has high fecundity, or it produces a large amount of seed per plant. Generally averaging, I would say, around 20 to 30,000 seeds per plant. But some research has shown ranging all the way up into the hundreds of thousands of seeds per plant. So that means if a plant has, say, for example, a mutation that results in resistance, you'll have 30,000, or you'll have a large number of those individuals entering into your seed bank and coming back in subsequent years. So it can select for resistance very quickly. But in addition to that, it has very low, it has almost no dormancy, no seed dormancy, which means that the seed bank in kochia is not persistent. It persists for generally about 1 or 2 years. And what that actually means, though, is that kochia, all of these seeds that are going into the seed bank, they tend to germinate within the either, say, next growing season or the growing season after. And if we have a new individual that has herbicide resistance, all of the progeny from that individual are crossing with one another and making the problem worse, allowing that population to evolve more rapidly compared to some of our other species. So
why is that?
Because there's resistance. Say you have one plant that's resistant, right? And you'll have, say, how many thousand individuals going into the seed bank, and those are coming back the next year, right? And that's more pollen to cross with other.
Good thing, because you can get, if you get rid of it in 2 years, you're free.
It is. So that's the one thing that you hear about kochia, right? So everywhere says the weak point in the life cycle of kochia is the fact that it does not persist for very long in the seed bank. But I do get where they're coming from with that, but with any population, there's always a very low number that persist for a very long period of time. So you can say reduce 90% of the population or more over if you manage the population completely for 2 years, right? But then you'll still have 10% of those 30,000 that can come back, right? So you have to be very diligent about managing these weed populations over and over again for many years in a row to try and actually deplete that seed bank fully. So you can deplete the seed bank quite effectively if you manage it for multiple years in a row, but that doesn't mean that all of the seeds are gone, right? So we've kind of covered a little bit about the biology of the weed, why it's so resistant. Another huge issue that we're finding is that you really, when you apply herbicides to kochia, you actually have to time it very well, because there tends to be kind of a certain stage in the life cycle that after you apply herbicides past that stage, they are not even close to as effective as they were before, right? So it can be quite easy to actually miss the correct stage to spray kochia. One of the other things that I wanted to talk about is that kochia, it's one of the first weeds to emerge in the spring. So it actually emerges after about 50 growing degree days here in Western Canada, meaning that most of the population has actually emerged before any of those other weeds you could see any of those other weeds out in the field. So one of the most important things for managing kochia is, first of all, using an efficacious pre-plant herbicide. So that means not just relying on glyphosate, but using multiple modes of action. A lot of our research has shown, I mean, the group 14s tend to work quite well for managing kochia as pre-plant herbicides. So you really have to be careful to include multiple modes of action and know if you have resistance out in your field, so you can include those multiple modes of action to basically get a head start on kochia. But then from there, kochia actually has what we call a prolonged emergence periodicity, which means that most of the population emerges the first thing in the spring, but then you have a small portion of that population that extends its emergence later into the growing season, to the point that you can have emergence after your in-crop or your post-emergence herbicide. Okay, so what we are advocating are layering pre-plant herbicides, but also post-emergence herbicides, and having multiple different modes of action that have activity on the kochia. So in addition to that, we've kind of covered a little bit on herbicide management of kochia. In the past, so the researcher that was in my position before me, Dr. Bob Blackshaw, he had led a previous round of research on kochia, and in that research they covered options for herbicide management. So he was working with a master's student who's actually working with Dr. Bill Hammond, and they tested the efficacy of a wide range of herbicides for managing kochia. Most of them were post-emergence herbicides. In addition, they also did some more ecology work, so looking at trying to answer the question of timing of emergence. So if you see this weed showing up at a certain time, say later and later in the growing season, can it still produce seed before the first fall frost? And what they actually found was that the plant can emerge up to the first few weeks in August. If it emerges, say, the first week in August, it could still produce viable seed before the end of the growing season. So it's very what we would call phenotypically plastic. It can adapt to a wide range of environments. So
what do you do here? Do you try to control this at all?
Yes.
Or is that the goal here, to keep it?
So, yeah.
Keep it viable?
That's a good question. So, yeah, moving on to some of the experiments that we have here. We're looking at a wide range of things, but the idea is that we're not only focusing on using herbicides, but we're also using what we would call cultural management, right? So this experiment behind me is looking at integrating, so this is a rotation experiment. It's a fully phased rotation. The rotation is spring wheat, canola, spring wheat, lentil. And what we mean by fully phased is that all of those phases or all of those crops in the rotation are present in this growing season. So we have that spring wheat canola, spring wheat lentil rotation, and we're growing that rotation using either narrow rows or wide rows. So in this case, our narrow row is 9 inches, our wide rows are very wide, they're 18 inches. And then we're also growing those rotations using a recommended seeding rate versus double the recommended seeding rate. So this is the experiment that you see behind me. This is the first year of that experiment. You're welcome to kind of walk up to the plots and look and see if you can spot any kochia in there. So the first thing I want to note is that these signs here, you'll see we do have signs explaining whether it's a recommended, let's say the 1x seeding rate or a narrow row spacing, for example. But then we also have the in-crop herbicides listed on there. These are actually signs showing what was done last year. But the idea is that in these rotations, if we have, say, a double seeding rate, narrow row spacing, it's double seeding rate, narrow row spacing in all of the crops in that rotation over the next 4 years.
What's the seeding rate?
So the seeding rates vary for the crops. I know I'm not sure in bushels per acre, but in seeds per meter squared. For the cereals, we're comparing 200 seeds per meter squared versus 400. For the canola, it is 100 seeds per meter squared versus 200. And for the lentil, it's 130 versus 260.
So the old seeding rate.
Pardon?
The old seeding rate.
Yeah.
So how many, over those 4 years, how many groups do you use?
How many herbicide groups?
How many herbicide groups do you use over the 4 years?
We're trying to use as many as we can, right? So the idea is there are different recropping restrictions for many of these herbicides, right? So the whole thing is looking at integrating what we're trying to come up with a best management practice from a herbicide rotation and mixing perspective, in addition with these cultural management tools. So I would say, you're really making me try and remember here. We would probably be using 5 or 6 modes of action over the course of a 4-year rotation. So in general,
Plus your number 9 or without that?
that's
Included.
that's included generally. So included with a lot of the pre-emergence herbicides, right? So looking at the plots, so behind me we're looking at a 1x narrow row seeding rate of canola. You can see the canola is kind of just poking through the surface here. So the in-crop herbicides that we're using in this one are looking at Liberty for an in-crop herbicide in canola, right? So our research in the past has shown that Liberty is quite effective actually for managing glyphosate resistant kochia. Whether you're using Liberty a single application in-crop or a double application. So note that these haven't actually received a post-emergence herbicide yet, but they have received a pre-emergence herbicide. So the pre-emergence herbicide used before canola was glyphosate plus AIM, or carfentrazone. Then we have a lentil plot over here. I think, yeah, lentils over here. So before the lentils, we would use what's called heat complete. So that would be saflufenicil plus pyroxasulfone. So this is the second year for this experiment. We're going into the second year right now. And I've actually found, this might not be the best example, but the lentil plots actually look fairly clean from that heat complete application at this point. So that is very nice, because lentil, we have very few options when you get into trying to manage kochia, because many of the most effective herbicides are group 2s. Yeah, exactly. Then we have spring wheat is another plot over there. And, oh, actually, so before spring wheat, it was glyphosate plus AIM, so glyphosate plus carfentrazone. Before canola, it was Edge, and then Edge, which would have been applied early spring this year, and then glyphosate application right before we planted the canola. So the idea is that if you can walk around, look at the signs. If it says 1x seed, that's the recommended seeding rate, and wide is the wide row spacing.
The last year's chemistry.
Last year's chemistry. So we're looking at. So this would have been spring wheat. So the other thing to mention with the spring wheat, due to recropping restrictions, right? So generally the herbicides that we would have used before canola, so that would have been an application of Infinity, which is bromoxynil plus, oh, I'm blanking now. It'll say on the sign. Yeah, so this would have been Buctril M because it's going into lentil. So that would be a group 6 for bromoxynil and then a group 4 for MCPA. But MCPA alone doesn't really have that great of activity on kochia. If we can find, what does this canola plot say over here? Yeah, so this would have been the Infinity application, which is pyrosulfatol, which is a group 27 herbicide, plus bromoxynil, which is a group 6, both of which we don't have resistance to that we know of yet. So when you get to growing spring cereals, Infinity is a very good option for kochia at this point because it doesn't have one of those group 4s that we're starting to see resistance to. Okay? So for the sake of time, you can kind of explore this experiment. I'm just going to start mentioning this one right beside us. So over here, there's no stakes or anything. This is the first year of another rotation study. So in this experiment, we're looking at crop rotation diversity, as well as the diversity in herbicide options that come along with that. So the rotations that are present in this experiment are, say, spring wheat lentil compared to a spring wheat canola rotation, compared to a spring wheat canola, spring wheat lentil rotation. But what we're doing with that, we have those 3 rotations with spring wheat, but then you can see right here, this would be winter wheat that was seeded last fall. So we have all of those 3 rotations replacing the spring wheat with winter wheat. So why would we do that?
To get ahead of the game, to get ahead in the spring.
Exactly. So the whole idea with this is that we're trying to alternate life cycles within our rotation, right? So if you grow all summer annual crops throughout your whole rotation, you'll select for weed problems that are summer annuals, right? So if we throw winter wheat into the rotation, the idea is that we're alternating those life cycles. We're having a canopy that's going to be closed a lot earlier in the growing season while that kochia is trying to emerge, right? So we're trying to basically compete with the kochia as much as possible. And the main measurement would be seed production of kochia in these plots.
No herbicide.
No herbicides.
They're not in plants either.
Pardon?
It's already ahead, so.
Yeah. So there is, I can see a kochia plant right there. But the idea is that those kochia plants aren't actually going to set seed, right? So kochia, it's the first weed to emerge in the spring, but it's also one of the last ones to die off. It waits for the first spring frost to cause the plant to start to senesce. But in addition to that, the seeds actually don't become viable, or there's a very low proportion of those seeds that are viable if you're harvesting early, or harvesting, say, a winter annual crop, right? So we're going to be cutting those plants off before they're going to be setting viable seed. And so if you compare this plot here compared to spring wheat, which is right beside it, you can see the obvious differences in how competitive this is going to be compared to that.
That's the normal time, right? The normal time is spring wheat.
Yes.
You didn't try to alter early, right? Yes.
No, we did not try, this experiment isn't looking at seeding date.
But you could. get
You
ahead.
could, yeah. So that's another interesting comment. So seeding date, one of the things that we're finding for herbicide-resistant kochia, specifically glyphosate-resistant or oxinic-resistant kochia compared to their susceptible counterparts, we're finding that there is actually a fitness penalty to the resistance. And what that means is that the plants that are resistant actually, without the application of the herbicide, they're not as fit to grow and produce seed and contribute to the next generation. So what we're seeing is that in general, the plants, they germinate slower, they emerge later, and what that means is that there's a potential, or we're seeing sort of a divergence in your herbicide susceptible kochia emerging very early, but then your herbicide resistant kochia starting to emerge later and later, meaning that it's going to be more difficult to catch that with your pre-seed application, right? But what that also means is that seeding date might be a very good option for managing kochia. If we can establish a crop very early in the spring, it will be more established by the time that herbicide-resistant kochia actually starts establishing within that crop, right?
How many types of kochia do we know of?
You're talking biotypes? There's a wide range. I don't have an answer for that though. I don't even know if that's characterized. I don't think it is.
I've heard 17 different types.
It's quite possible, yeah. So there's, in addition to all of these biotypes that you're seeing out here, there's also ornamental biotypes, right? So kochia, it originated in Europe and Asia, and it was brought in as an ornamental originally, but then later on brought in as a forage crop. So for the sake of time, we're going to continue on down back to the trucks there, and there's another experiment that I'd like to quickly cover as well. Are there any questions on this? Not hearing any?
Like you're saying, as you're walking, you can tell the different types of kochia.
Yeah.
But which one are they and which one's going to be resistant?
Exactly, yeah.
We see that all the time in the field. That one's dead, that one's not, that one is. Then the customers are complaining like they didn't kill them.
Exactly, yeah. So the other thing to consider with herbicide resistance is that it's generally only present in a portion of that weed population, right? So when we do our testing, we can see, say, glyphosate resistance, for example, in kochia showing up in 1% of the population, or all the way up to pretty much the entire population, right? So there's a proportion of these individuals that are resistant, but also a proportion that are susceptible. So this isn't a kochia experiment here actually, but while we're walking past, this is a 12-year rotation here, looking at frequency of canola and crop rotations. It's going into its 12th year this year. So we're comparing a continuous rotation of canola, so canola every year, versus a canola wheat rotation versus a canola pea barley rotation. So you can kind of tell if you look, there's no stakes here, but if you look at the stubble, you can kind of tell which rotation it is. But if you see canola on canola stubble, that's canola that's been grown on canola stubble for 12 years in a row. Yeah, so this experiment, that experiment was actually led out of Lacombe by Dr. Neil Harker. And Brienne's taking it over. And what the results after 6 years showed that you do see that progressive yield decline as those rotations get tighter. And it was mainly due to a buildup of blackleg and also root maggot. So this is kind of a weed garden here. What we're looking at, we have kochia seeded in these plots here. And we're actually looking at what I'm going to call the aftermath of last year. Okay, so if you look at the signs, what this experiment is looking at is cutting timing of kochia populations, and as well as integrating that with pre-harvest or post-harvest herbicides. So why would we be interested in cutting timing? Does anyone know?
Prevent it from seed setting.
Exactly. So first thing we're trying to do is trying to answer the question, when does the seed on a kochia plant become viable, right? Surprisingly, we don't know that yet. So we're cutting these populations off every 2 weeks. We start on basically the 1st of August, and then progressively every 2 weeks we go into mid-October. and we're looking at viability of that seed and how much seed is produced.
Yeah,
The
but
re-
it wouldn't produce seed into October, no?
The kochia is really interesting, right? So it's very indeterminate, which means that if the conditions are there, it will produce seed. So I have some really interesting pictures actually from this experiment last fall, where we have kochia up to here. It was probably the start of October, we had a snowfall. There's snow covering those plants, but as it's melting, you can actually see the tips of the weeds start growing again. So you have mature seed throughout the plant, and then it's starting to grow and produce new seed in October.
We gotta find a use for it.
Yeah.
So we
Exactly, yes. So speaking of a use for it, okay, the other objectives in this experiment, it's kind of based around, a lot of times we know kochia remains green very late into the growing season, right? So usually when you would go in and your normal time for harvesting a summer annual crop, the kochia would still be green, and that creates a lot of difficulty trying to put that weed through your combine, right? So a lot of times that really almost makes it necessary to use either swath it down or use a pre-harvest herbicide, right? So what this is looking at, it's looking at timing of cutting those populations down, as well as if we use a pre-harvest herbicide, is that going to impact the viability of that seed before you cut it down as well? In addition to that, we're looking at using a post-harvest herbicide as well. So after you cut the population and use a post-harvest herbicide, in this case it's glyphosate plus heat, will that stop the population from regrowing and trying to produce more seed? Okay.
But your resistance you talked about earlier was to stay green, right? To stay green.
What do you mean?
When you spray glyphosate, it was stay green, it wouldn't die.
Yeah.
Okay.
Yeah.
So,
So
with the population becoming glyphosate tolerant, is there any point in doing,
there is, yeah. So, well, I can't say that there is and that there isn't. That's what we're trying to determine, right? So we do know that that using, say, a herbicide combo like glyphosate and heat on glyphosate-resistant kochia can actually get it to dry down, making, say, a direct harvest easier, right?
But doesn't die?
But it doesn't die. So you can wander around here. You can even walk in this area of the plots if you want. But what you want to compare, we have on the signs, we have either pre-harvest for a pre-harvest application of glyphosate plus heat. We have post-harvest for a post-harvest application of glyphosate plus heat, or the untreated controls where we just went out and cut the populations off. Then we have cut dates 1 through 6, getting progressively later, 1 being cutting the start of August, and then progressively every 2 weeks. So cut date number 6 would have been a population that was left growing until about mid-October, and we would have cut it off at that point. So you can,
Dandelion control is one of the side effects?
pardon?
Dandelion control is just one of the side effects?
Yes. So actually, there's something really interesting, at least to me here. If we go down, let's go down a couple plots down here. So if we look at, if we're comparing these plots right here, I'm assuming these are later cutting dates.
Cut 2.
Oh, this is cut 2,
This is
6. So what I was wanting to show here is that we have essentially a mat of kochia on the surface, right? And if you go farther down there, you can see that there's plots where the kochia looks a lot more green. It's really thriving compared to these plots where it's almost looking like it's suppressed, right? And so this it's not an effect of the herbicide. It's actually the allelopathic potential of kochia. So this is what we call autotoxicity, meaning that the kochia from last year or the biomass from last year and the density of seedlings that are growing this year, they're actually inhibiting their neighbors. So from that, what we can kind of say is that if the problem gets bad enough, it'll manage itself. But we don't know if a crop would actually grow here or not, right? So So we actually have an area over there where it's much worse. And that
You ever tried dicot on it?
dicot?
Like for if you want to really control it?
Yeah, so I haven't tried it in this research project, but I know that it does tend to, it's one of the better ones as far as managing it later on in the season. Yeah. So you can walk up and down here and compare the cutting dates. I kind of took a quick walk down here before you guys got out here. And what I thought I was seeing was that if you look at cutting dates 3 or 4 in general, you tend to see less kochia emerging this spring compared to the early cutting dates or the late cutting dates. So why would anyone know why that would be?
Because the kochia was not long far enough to make seed, but it was far enough to have flower. So
Exactly, yeah. So the idea is that if we cut the kochia late in the growing season, it's already produced viable seed, and that seed is falling off the plant as you're cutting it and going into the seed bank, making basically a more dense population of kochia for next year, right? So harvesting late may not be a good idea, or cutting kochia late. And conversely, if we look at early cutting dates, we'll be cutting the kochia plants before that seed becomes viable, but actually while the kochia is still vegetative. And if you cut early, it actually has enough energy to come back and put all of that energy into producing as many seeds as possible before the end of the growing season, right? So if you cut early, you have regrowth at the start, and then as you start cutting later, there's less and less regrowth. But also, if you do see the viability of the seed increasing as you get to those later and later cutting dates.
So what was 3 and 4 be? September 1st or something like that?
I have that written down here somewhere.
I mean, there wouldn't be a point where it wouldn't quit
It
trying to make viable seed.
well, it quits after you get the first fall frost, which causes it to senesce.
Well, I would have to
Yeah.
be the weather.
So 3 and 4 is September.
So
That's horrible stuff.
So yeah, the cut date 3 would be the very end of August, start of September, and then cut date 4 is the second week of September, which is kind of good news because that's when a lot of the harvest happens, right? A lot of harvest of summer annual crops. So in addition to that, I know I'm kind of going a little bit over time here, but the implications for this project are looking at patch management as well, right? So a lot of times we have these green patches of kochia in the low-lying areas of the field, and many growers know that they're not going to be able to harvest through those green patches. So often what they do is they harvest around them, and they leave them out in the field until they're done harvest, right? And all the while the seed on the plant is becoming more and more viable. So we want to know how late can we actually leave those patches before we come in and cut them. And usually when they cut those patches off, the kochia in the past has actually been bred as a forage crop. So it has fairly good actual feed value, similar to alfalfa. So a lot of growers are feeding these, they're basically baling it up and using it as feed for cattle. But we do know from previous research that seed can actually, some of that seed can remain viable through the digestive tract of an animal. So ruminant digestion does not fully decrease viability in this plant.
So bring the harvest swath out at the same time as harvest.
That's the idea, yeah. So swathing at the time of harvest would actually work quite well. At least that's what our preliminary results are showing. We've only done this in Lethbridge. We had 2 site years of this in Lethbridge this past year. Then this summer, we have 2 sites in Lethbridge. Then we have 1 site in Scott, Saskatchewan. We have 1 site in Saskatoon, Saskatchewan, as well as 1 site in Carmen, Manitoba, and 1 site in Winnipeg, Manitoba. We've only done this for the 1 year so far. We're going into our 2nd year, but what you're seeing are the leftover effects of the 1st year. Yeah. So are there any questions at this point?
I was curious about that feed value thing. Is there a happy medium where you can still cut for feed and not have those viable seeds produced at the same time?
Well, that's what I'm hoping to determine with this. So initially, we weren't looking at the feed value of the kochia at these different cutting dates, but I do hope to add that in for this year to look at basically those feed value parameters to see how that progresses as these cutting dates get later and later.
Because you can't beat it jointed in a way. Is it almost the way that you have to think about it?
Yeah,
Right?
exactly. I do know, so as it gets into the reproductive stages, it does start to get really woody, right? Meaning that there's more fiber included in there. But yeah, so we don't know that yet, but we're hoping to answer that.
My understanding is that there's been no modes of action in herbicides discovered in the last 10 to 20 years.
Almost 30.
Is that basically true? And if so, what is being done in the general research community regarding that?
Okay, that's a big question.
The biggest question.
Okay, I'm gonna say, because we're running out of time, this is gonna be the last question I'm gonna answer, but come up and talk to me afterwards if you have more. So no new modes of action have been released in the high 20s, going into almost 30 years now, right? With that said, I've heard that there may be 1 or 2 modes of action coming down the pipeline. I have no idea when that will be. It won't be anytime soon. And we don't know, there's always so many implications.
RNAi, what about that?
Yeah, so RNAi, personally, I mean, in the research community, there seems to be multiple different opinions on RNAi. I do believe that RNAi could be a viable option, especially for the type of resistance, the type of glyphosate resistance that we have in kochia, right? So RNAi, very broadly in general, it suppresses genes, right? So you can use it to suppress genes. And the type of resistance that we have in kochia is what's called gene amplification or target site amplification. So when a herbicide tries to manage a weed, right, it binds to a target site, right? But these kochia plants, they're resistant. They have multiple target sites, right? So they have more than one, meaning that you need higher and higher rates of the herbicide to the point now that you can't spray rates that high. But the idea being that RNAi could possibly be used to reduce the number of viable target sites in the plant, making glyphosate effective again. That's a hypothesis. It's not been proven, so. Anyway, so I think we're out of time, right?
You betcha.
Okay.
Join me in thanking Charles. Charles, since you have the mic on, can you explain where shop